Section 12.6 Properties of Solutions. The solute particles in a solution play an important role in determining the properties of the solution. In most of the solutions discussed so far, the solute is dissolved as small particles that are uniformly dispersed throughout the solvent to give a homogeneous solution. When you observe a solution, such as salt water, you cannot visually distinguish the solute from the solvent. The solution appears transparent, although it may have a color. The particles are so small that they go through filters and through semipermeable membranes. A semipermeable membrane allows solvent molecules such as water and very small solute particles to pass through, but does not allow the passage of large solute molecules. Colloids. The particles in a colloidal dispersion, or a colloid, are much larger than solute particles in a solution. Colloidal particles are large molecules such as proteins, or groups of molecules or ions. Colloids are homogeneous mixtures that do not separate or settle out. Colloidal particles are small enough to pass through filters, but too large to go through semipermeable membranes. Table 12.9 lists several examples of colloids. So, fog and smoke and shaving cream and mayonnaise and gelatin, clouds, milk, these are all colloids. Freezing point lowering and boiling point elevation. When a solute is added to water, the physical properties such as freezing point and boiling point change. Therefore, an aqueous solution will have a lower freezing point and a higher boiling point than pure water. These types of changes in physical properties, known as collocative properties, depend upon the number of solute particles in that solution. Probably one familiar example in the process of spreading salt on icy sidewalks and roads when temperatures drop below freezing. The particles from the salt combine with water to lower the freezing point, which causes the ice to melt. Another example is the addition of antifreeze, such as ethylene glycol, to water in the car radiator. Ethylene glycol is an organic compound with two alcohol functional subgroups, which form hydrogen bonds, which makes it very soluble in water. If the ethylene glycol in the water mixture is about 50-50 by mass, it does not freeze until the temperature drops to about -30 Fahrenheit and doesn't boil unless the temperature reaches 225 Fahrenheit. The solution in the radiator prevents the water in the radiator from forming ice in cold weather and boiling over in hot desert highway. Insects and fish in climates with sub-freezing temperatures control ice formation by producing biological antifreezes made of glycerol, proteins, and sugars such as glucose within their bodies. Some insects can survive temperatures below -60 Celsius. These forms of biological antifreezes may one day be applied to long-term preservation of human organs. Particles in solution. A solute that is a non-electrolyte dissolves as molecules, whereas a solute that's a strong electrolyte dissolves entirely as ions. The solute is antifreeze, which is ethylene glycol, a non-electrolyte, dissolves as molecules. Molality. The calculation for freezing point lowering or boiling point elevation uses a concentration unit of molality. The molality, abbreviation italicized M, of a solution is the number of moles of solute per kilogram of solvent. This may seem similar to molarity, but the denominator for molality refers to the mass of the solvent, not the volume of the solution. Using molality to calculate freezing point lowering and boiling point elevation. 1 mole of particles in 1,000 grams of water lowers the freezing point from 0 to -1.86 Celsius. A similar change occurs when the boiling point of water. 1 mole of particles in 1,000 grams of water raises the boiling point from 100 to 100.52 Celsius. The freezing point lowering, delta Tf, is determined from the molality of the particles of the solution and the freezing point constant, K sub F. So delta T, the change in temperature, is equal to the molality times the freezing point constant. We can calculate the new lower freezing point as temperature of solution is the temperature of solvent minus the change in temperature. The freezing point constant for water is 1.86 degrees Celsius over molality. If there are 2 moles of particles dissolved in 1 kilogram of water, the freezing point decreases twice as much to negative 3.72. For example, the solution of 1 mole of sodium chloride in 1 kilogram of water produces 2 moles of particles, 1 mole of sodium and 1 mole of chlorine ions. Thus, 1 mole of sodium chloride in 1 kilogram of water produces a 2 molal solution of particles. A similar change occurs with the boiling point of water. The boiling point elevation, delta T sub B, is determined from the molality times the boiling point constant. Osmotic pressure. The movement of water into and out of the cells of plants as well as our own bodies is an important biological process that also depends on solute concentration. In osmosis, a semipermeable membrane allows molecules of the solvent, water, to move through but retains the solute molecules. In this process of diffusion, water moves from the compartment where the concentration is higher to the side where it has lower concentration. In terms of solute concentration, water flows through the membrane in the direction that will equalize or attempt to equalize the concentrations of solute on both sides. Although water can flow in both directions through a semipermeable membrane, the net flow of water is from the side with the lower solute concentration to the higher solute concentration. If an osmosis apparatus contains water on one side and sucrose solution on the other side, the net flow of water will be from the pure water into the sucrose solution, which increases its volume and lowers its sucrose concentration. In the case where 2 sucrose solutions with different concentrations are placed on each side of the semipermeable membrane, water will flow from the side containing the lower sucrose concentration into the side containing the higher sucrose concentration. Eventually, the height of the sucrose solution creates sufficient pressure to equalize the flow of water between the two compartments. This pressure, called osmotic pressure, prevents the flow of additional water into the more concentrated solution. Then there is no further change in the volumes of the two solutions. The osmotic pressure depends upon the concentration of the solute particles in the solution. The greater the number of particles dissolved, the higher the osmotic pressure. In this example, the sucrose solution has a higher osmotic pressure than pure water, which has an osmotic pressure of 0. In a process called reverse osmosis, a pressure greater than the osmotic pressure is applied to a solution so that it's forced through a purification membrane. The flow of water is reversed because water flows from an area of lower water concentration to an area of higher water concentration. The molecules and ions of the solution stay behind, trapped by the membrane, while water passes through the membrane. This process of reverse osmosis is used in a few desalination plants to obtain pure water from seawater. However, the pressure that must be applied requires so much energy that reverse osmosis is not yet an economical method for obtaining pure water in most parts of the world. Isotonic solutions. Because the cell membrane and biological systems are semipermeable, osmosis is an ongoing process. The solutes in body solutions such as blood, tissue, lymph, and plasma all exert osmotic pressure. Most IV solutions, intravenous, are used in hospitals are isotonic solutions, which exert the same osmotic pressure as body fluids such as blood. The percent concentration typically used in IV solutions is similar to the types of percent concentrations we've already discussed, except that the concentration of IV solution is mass over volume percent. The most typical isotonic solution is 0.9% mass to volume. Sodium chloride, or 0.9 sodium chlorides per 100 ml of solution, and 5 of glucose solution, or 5 grams per 100 ml of solution. Although they do not contain the same kinds of particles, the glucose solution are both 0.3 molar sodium and chloride or glucose molecules. A red blood cell placed in an isotonic solution retains its volume because there's an equal flow of water into and out of the cell. Hypotonic and hypertonic solutions. If a red blood cell is placed in a solution that is not isotonic, the differences in osmotic pressure inside and outside the cell can drastically alter the volume of the cell. When a red blood cell is placed in a hypotonic solution, which has a lower solute solution, water flows into the cell by osmosis. The increase of fluid causes the cell to swell and possibly burst, a process called hemolysis. A similar process occurs when you place dehydrated food, such as raisins or dried fruit, in water. The water enters the cells and the food becomes plump and smooth. If a red blood cell is placed in a hypertonic solution, which has a higher solute concentration, water flows out of the cell into the hypertonic solution by osmosis. Suppose a red blood cell is placed in a 10% sodium chloride solution. Because the osmotic pressure in the cell is equal to that of 0.9 sodium chloride solution, the cell shrinks, a process called crenation. A similar process occurs when making pickles, which uses a hypotonic salt solution that causes the cucumbers to shrivel as they lose water. Dialysis is a process similar to osmosis. In dialysis, a semipermeable membrane called a dialyzing membrane permits small solute particles and ions as well as solvent water to pass through, but it retains large particles such as colloids. Dialysis is a way to separate a solution from its colloids.